Welcome to the second part of Module 5. In this module, we will focus on the different types of biological molecules. The learning outcomes for the second part of Module 5 are: identify the different biological molecules, determine the monomers and polymers for biological molecules, and identify the main functions for these biological molecules. Biological molecules can be classified as inorganic compounds or organic compounds. Inorganic compounds generally lack carbon, with a few exceptions, and they have a simple structure. The bonds between the elements are either ionic or covalent. Examples include water, salt, hydrochloric acid, and sodium hydroxide. Carbon dioxide and the hydrogen carbonate ion both contain a carbon atom, but they have simple structures and behave like inorganic compounds. Organic compounds always contain carbon, and most of them will also have hydrogen. The elements in these compounds are bonded by covalent bonds, and they can have a very complex structure. First, let's start with some definitions. Monomers are the single units or building blocks for polymers. Polymers are made of repeating units of monomers. The term macromolecules refers to very large organic molecules, polymers, that form from combining small organic molecules, the monomers. Macromolecules such as carbohydrates, lipids, proteins, and DNA, or deoxyribonucleic acid, are generally synthesized through dehydration reactions, which is the loss of water. The first group of organic compounds we will cover are the carbohydrates. Carbohydrates are a large and diverse group of organic compounds. Carbohydrates contain carbon, hydrogen, and oxygen, and the common ratio is CH2O. So there are 2 hydrogen atoms for every carbon atom. Also, there is 1 oxygen atom for every carbon. So if we have a sugar molecule with 6 carbons, then we find 12 hydrogens and 6 oxygens. They are very important in our diet as they provide lots of energy to our body. Carbohydrates have other important functions as well. Some examples of carbohydrates include glucose, which is a very important monosaccharide. Glucose is the sugar in our blood, and it is the fuel for our brains. Sucrose is table sugar. It is a disaccharide. Glycogen is a polymer or polysaccharide that contains many glucose units. Other examples are starch, which is a storage form of carbohydrates in plants, and cellulose, which is present in plant cell walls. The main function of carbohydrates is to provide energy to generate ATP molecules. ATP, as you studied previously, is the main fuel source for processes in our body. When ATP supplies are sufficient, dietary carbohydrates are converted to glycogen or fat. Carbohydrates are also important for structural purposes. For example, DNA contains deoxyribose, which is a sugar. RNA also contains ribose, which is also a sugar. Carbohydrates can be classified according to their size into monosaccharides or monomers, which means 1 unit of sugar, like glucose, disaccharides, which have 2 sugar units bonded together, like sucrose, or polysaccharides, which are the polymers, which means they have many sugar units bonded together. Sucrose is a disaccharide which is made of a glucose unit and a fructose unit. Notice that both glucose and fructose have the same molecular formula of C6H12O6, but they are 2 different sugars, as their structures are different due to their different atom arrangements. So those 2 units come together in a condensation reaction. The 2 sugar units are bound together by a covalent bond known as a glycosidic bond. The reverse reaction requires hydrolysis. So adding a water molecule to the sucrose breaks the glycosidic bond and separates the sucrose into glucose and fructose again. As you can see, carbohydrates contain many OH groups, so they are polar compounds and thus they dissolve in water. Glycogen is a polymer and it is the storage form of glucose in the body. From the illustration you can see that the glucose units bonded together through the glycosidic bond. The second type of organic compounds are the lipids. In terms of the chemical structure, lipids also contain carbon, hydrogen, and oxygen. This is an example of a triglyceride. You can see there are around 50 carbons and around 100 hydrogens, but only 6 oxygens. So compared to carbohydrates, lipids contain lots more carbon, lots more hydrogen, but less oxygen. So these compounds are non-polar or hydrophobic, so that's why oil and water don't mix. In lipids, we will not discuss monomers or polymers, as there aren't any common examples. What is common between all lipids is that they are hydrophobic and have different roles in the human body. Fatty acids and triglycerides are the two types of lipids that our body stores when we eat excess food. Phospholipids are the major component of the cell membranes. You can see from the table the wide range of functions that lipids play in our body. Let's discuss the fatty acids and the triglycerides in more detail. Fatty acids have a simple structure. You can see many carbon-carbon and carbon-hydrogen bonds. At the end of the structure, there is a COOH, which is called a carboxylic acid functional group. This structure explains the name of the molecules. So the part of the molecule that contains carbons and hydrogens gives the name fatty. This part is non-polar. The carboxylic functional group gives the name acid, and this part is polar or hydrophilic. You can see that the structure of this fatty acid is linear, so molecules can stack closely together. You can also see that we represent the molecule by drawing lines instead of writing all the carbons and all the hydrogens. Fatty acids are used to synthesize triglycerides and phospholipids. There are two types of fatty acids, saturated or unsaturated. Saturated fatty acids means that all the carbon-carbon bonds in the fat are single bonds, so each carbon atom is saturated with hydrogen atoms. Unsaturated fatty acids mean at least one carbon-carbon bond is a double bond, so the carbon on the side of the double bond are not completely saturated with hydrogens. From the structure here, you may notice that the double bond causes a kink in the structure of the fatty acids. This kink prevents the molecules from stacking together like they do in saturated fatty acids. This will impact the physical properties of the fatty acid. This is another illustration of saturated and unsaturated fatty acids. If the hydrocarbon chain has one carbon-carbon double bond, then the fatty acid is monounsaturated. If there are more than one double bond, the fatty acid is polyunsaturated. You can also see that there are two configurations that the double bond can cause in unsaturated fatty acids. In the first example, the two hydrogen atoms are located on the same side, and the carbon chains on the other side. This configuration is called cis. The other configuration of the double bond is called trans. In this configuration, the two carbon chains are on opposite sides of the double bond. The trans fats are very unhealthy for our body, and we should avoid products that contain them. Unsaturation of the fatty acids lowers the melting point. This is because the double bond puts a kink in the hydrocarbon chain, preventing the chains from packing closely together and solidifying. As such, the fatty acids of a fat, which is solid at room temperature, are mostly saturated, whilst the fatty acids of an oil, a liquid at room temperature, are mostly unsaturated. For example, stearic acid is a saturated fatty acid that is found in animals. It has a melting point of 70 degrees. Oleic acid is monounsaturated. The double bond causes a kink which prevents molecules from stacking together, so it has a lower melting point of 14 degrees. This fat is the major component of olive oil. When three fatty acids come together and bind to one molecule of glycerol through condensation, they form triglycerides. The linkage between the fatty acid and the glycerol is called an ester linkage. Triglycerides are fat storage in our body. They can be either solid like lard or liquid like olive oil. The saturation level of the fatty acid is the reason for that. So triglycerides with saturated fatty acids have a higher melting point, and they are normally solid at room temperature. On the contrary, triglycerides with unsaturated fatty acids have low melting points, and they are liquid at room temperature. The 3-carbon glycerol forms the backbone of the triglycerides, forming an E-shaped molecule. The glycerol backbone is the same in all triglycerides, but the fatty acid chains vary, resulting in different kinds of fats and oils. Phospholipids are the major component of the cell membrane. They have a very similar structure to the triglycerides. They consist of a glycerol backbone that is bonded to only 2 fatty acids. The 3rd carbon of the glycerol has a phosphate group, which is polar, so it is able to interact with water. The phospholipid structure has the hydrophobic tails formed from the fatty acids and a hydrophilic head that consists of the polar head. Cell membranes consist of a phospholipid bilayer, where the polar heads are pointing outside the cell and inside the cell so that they can react with water. The hydrophobic tails are facing inside the bilayer. Steroids are another example of lipids. They have different structure compared to the triglycerides. Steroids have a general structure of 4 rings. Different steroids vary in the arrangement of the atoms around the 4-ring structure. Some examples of steroids include cholesterol, estrogen, testosterone, cortisol, vitamin D, and some bile salts. 3. Steroids consist of many carbon and hydrogen atoms, but very few oxygens, so these compounds are also non-polar. The most important steroid is cholesterol, which we ingest in eggs, meat, and cheese. Our liver also produces cholesterol. Because lipids are non-polar, they don't dissolve in water or blood. For example, cholesterol needs lipoproteins to transfer it around the body. So when we consume too many lipids, they start to clump together and form plaque that clogs our arteries. Proteins are made of carbon, hydrogen, and oxygen like carbohydrates and lipids, but they also contain nitrogen, sulfur, and phosphorus. One example of the largest human protein is called titin, which contains 169,000 carbons and 270,000 hydrogens, as well as other atoms, so it is a very large and complex molecule. Proteins are structurally much more complex than carbohydrates or lipids and have many vital roles in cell function. Amino acids are the monomers of proteins, meaning they are the building blocks for proteins. There are 20 common amino acids. The generic structure for amino acids are 3 groups attached to a central carbon. The groups are an amino group, a carboxylic acid group, and an R group. The R group represents the side chain, and there are 20 different side chains, hence the 20 different types of amino acids. The amino group has a basic property, so it can accept a proton. The carboxylic acid groups has acidic properties, so it can donate a proton. Therefore, the generic structure of the amino acid can be written as doubly ionized form, which means that the carboxylic acid group donated the proton to the amino group. The amino acids can be classified according to the nature of the R group as nonpolar, when they contain lots of carbons and hydrogens, polar, when they contain OH groups, or charged, when they have either a positive or negative charge. The nonpolar amino acids are hydrophobic, so they avoid water. The polar and charged amino acids are hydrophilic, and they interact with water. A protein is synthesized by joining amino acids through condensation reactions, with the amine of one amino acid linked to the carboxylic acid of the next amino acid. The amino acids are linked together through a peptide bond. 2 amino acids linked together form a dipeptide. 3 amino acids linked together form a tripeptide. 10 or more form a polypeptide. The reverse reaction requires hydrolysis of the peptide bond to break the peptide into amino acids. Proteins contain at least 50 amino acids. During digestion, we hydrolyze the protein to break it down into amino acids that we use in our body. The following table provides the important functions of proteins. Proteins have important structural, regulatory, immunological, and transport roles. Proteins also have a catalytic role as enzymes. The structure of proteins consists of 4 levels, and we will look into each level. Primary structure is determined by the sequence of amino acids linked together by the peptide bonds. The primary structure is the backbone of the protein molecule. The secondary structure has 2 types, alpha helixes and beta sheets. The secondary structure is formed as a result of hydrogen bonds that stabilize this structure. The alpha helix has a structure similar to a phone cord. Beta sheet looks more like a corrugated roof or accordion bellows that goes up and down. The tertiary level is the overall 3-dimensional shape of the protein. Each protein has a unique tertiary structure that determines its function. A quaternary structure happens in some proteins when there are 2 or more protein strands that will interact together. A famous example is hemoglobin, which has 4 subunits that interact together to provide the quaternary structure. As we mentioned previously, enzymes are proteins that regulate chemical reactions by acting as catalysts. Some examples of enzymes include amylase, sucrase, and ATPase. Notice the suffix ase, which indicates that this protein is an enzyme. Chemical reactions occur when bonds break or form from the collision of atoms, ions, or molecules with one another. We can run the reaction faster by heating up, so the molecules have more energy and collide with each other with more force. We can do this in the lab, however, we cannot do that in our own bodies, as we have a constant temperature of 37 degrees. So we need enzymes to regulate the reactions in our body by acting as catalysts and lowering the activation energy. This is an example of a catalyst that is involved in the hydrolysis reaction of sucrose into glucose and fructose. The enzyme sucrase has a 3-dimensional structure with a space where the sucrose and water fit in. This space is called the active site of the enzyme. The The sucrose and water bind to the active site of the enzyme and undergo hydrolysis, so the sucrose is hydrolyzed by the water and breaks into glucose and fructose. These products then free the active site of the enzyme so it can be involved in the next reaction. As we saw in the previous slide, catalysts are not used up in the reaction, so a single catalyst can repeatedly catalyze one reaction after another. Enzymes are classified according to the type of reaction they catalyze. For example, oxidases add oxygen, lipases break down lipids, dehydrogenases remove hydrogen, and anhydrases remove water. A substrate is a term that refers to the reactants. So substrate means the same thing as reactants. The three most important properties of enzymes are: enzymes are highly specific. Each enzyme binds only to a specific substrate in the enzyme's active site, temporarily forming an enzyme-substrate complex. For example, maltose is a disaccharide that has a different structure from sucrose, so maltose will not fit in the active site of the sucrase enzyme. Enzymes are efficient. First, they don't get used up in the reaction, and second, under optimal conditions, enzymes can catalyze reactions up to 10 billion times faster than the reaction would proceed in the absence of them. Enzymes are subject to cellular controls. Enzyme activity is controlled by the chemical environment within the cell. Nucleic acids are the last biological molecule that we will cover in this module. Nucleic acids are the largest molecules in the body and are composed of carbon, hydrogen, nitrogen, oxygen, and phosphorus. There are two classes of nucleic acids: deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA. The relationship between DNA, RNA, and proteins is described in the central dogma of life, which shows how DNA, RNA, and proteins are related to each other. The building block for nucleic acids are the nucleotides, which consist of nitrogen-containing base, a pentose sugar, and a phosphate group. In DNA, the nitrogen bases are adenine, thymine, cytosine, and guanine, and the pentose sugar is deoxyribose. The carbons in the pentose sugar are numbered as 1', 2', 3', 4', and 5'. The sugar is called deoxy, which means it is missing an oxygen atom on carbon 2'. The structure of DNA is a double helix resembling a spiral ladder. The phosphate groups and deoxyribose sugars form the uprights of the ladder, and the bases form the rungs. We talked about the alpha helix being one form of a protein's secondary structure. Now we mention the double helix structure of DNA. So what is the difference between an alpha helix and a double helix? An alpha helix is one type of the protein's secondary structure. It consists of a single strand which is stabilized by vertical hydrogen bonds. The double helix forms the DNA structure, and there are two strands which are stabilized by horizontal hydrogen bonds. Remember that for a hydrogen bond to occur, we need the delta positive hydrogen from one molecule and the delta minus atom from another. In adenine, there is a hydrogen that is covalently bonded to a nitrogen facing an oxygen atom from thymine. So this is the first H bond. In thymine, there is a hydrogen that is covalently bonded to a nitrogen facing a nitrogen atom from the adenine. So this is the second hydrogen bond. The two structures of adenine and thymine align in this manner to allow for hydrogen bonds to happen, and the structures are called complementary. Similarly, there are three hydrogen bonds between guanine and cytosine, and they are also complementary bases. We can represent the two poles of hydrogen bonds as blue and red circles, where red means partially negative charged atom and blue means a partially positively charged hydrogen. As you can see from this diagram, thymine is not able to bind to guanine as the structures don't allow for hydrogen bonds because atoms carrying the same charge will face each other, so the atoms will repel. RNA has a very similar structure to DNA. However, there are three major differences between RNA and DNA. RNA is single-stranded, and the sugar is ribose, so it has an oxygen on carbon 2'. And RNA contains uracil instead of thymine. Adenosine triphosphate, or ATP, is the energy currency in our body. The synthesis of ATP happens due to the energy released from the catabolism of glucose. ATP has very similar structure to the RNA nucleotide adenine. The difference is that there are 3 phosphate groups linked together. If we remove the last phosphate group, then we get ADP, which means adenosine with 2 phosphates. If we remove another phosphate group, then we get AMP, which means adenosine with 1 phosphate group only. You only need to learn this simple structure of ATP for this course. The energy is stored within the bonds between the phosphate groups. So when ATP is hydrolyzed, it releases energy, producing adenosine diphosphate, ADP, and a phosphate group. ADP can be recycled to produce ATP again using the enzyme ATP synthase, and energy coming from the breakdown of glucose. This is the end of the Module 5 content. In the next few slides, we will summarize the key points in Module 5. This table provides a summary of ionic and covalent bonds. In Part 1 of Module 5, we reviewed the ionic and molecular bonds and the electronegativity. We also introduced the hydrogen bond as forces of attraction between 2 molecules. There are 4 criteria for the hydrogen bond to occur. Firstly, we need 2 molecules. Secondly, 1 molecule should contain hydrogen that is covalently bonded to either fluorine, oxygen, or nitrogen. The hydrogen will carry a partial positive charge. The second molecule should contain either oxygen, nitrogen, or fluorine atom with a partial negative charge. The negative and positive charges will get attracted to each other, and a bridge, a hydrogen bond, will occur between the 2 molecules. Some examples where hydrogen bonds occur include in water molecules, within the secondary structure of proteins, and in the double helix of DNA. After that, we covered the 4 types of biological reactions. Synthesis is to build larger molecules, and it is called anabolism. Decomposition is to break down molecules into smaller components, and it is called catabolism. Exchange reactions are very popular, and they consist of both synthesis and decomposition reactions. There are 2 examples of exchange reactions. The hydrolysis, which refers to breaking down molecules with the addition of water, and condensation, which refers to building up molecules with the loss of water. The last type of biological reaction is the reversible reaction, which occurs in 2 ways. We also introduced the inorganic acids as proton donors and inorganic bases as proton acceptors. In the second part of Module 5, we covered the biological molecules. First, we introduced the carbohydrates and said the monomers are the monosaccharide, such as glucose, fructose, and ribose. The monomers can join together in a condensation reaction to form polymers. The link between the monomers is called a glycosidic bond. Some of the important functions of carbohydrates are storage of energy and structural support. The second type of biological molecules that we covered were lipids. There isn't a single monomer that is common to all structures of lipids. For example, the structure of triglycerides is very different from steroids. Some of the important functions for lipids include energy and cell membrane structure. After that, we investigated the proteins. The monomers of these are amino acids, and there are 20 different types. They join together by condensation reactions to produce proteins, and the link is called a peptide bond. Some of the important functions of proteins include transport of oxygen by hemoglobin and the reaction catalysis by enzymes. Finally, we discussed the nucleic acids. The monomers are nucleotides, and there are 5 of them. They join together to produce either DNA or RNA polymers. They store and transmit hereditary information. Also, ATP is the energy currency in our body. This is the end of Module 5. Please practice the weekly exercises in your textbook and the questions you find in your workbook. We will meet face-to-face in the tutorial session, and this will be your opportunity to ask any questions.